This method demonstrates the calculation of quantifying protein amount in liquid sample with single molecule fluorescence spot counting. Specifically, it can elucidate how protein populations can be fluorescently labeled at each molecule level. Previous methods performed bulk measurements to analyze molar ratios of fluorescent and protein molecules, but they cannot reveal how heterogeneously protein populations are actually labeled. Our method can do it directly at the single molecule level. Our method can be extended towards ultrasensitive and advanced molecular concentration assays, which will lead to future diagnostic methods, allowing efficient findings of pathogenic molecules in specimens. Our method can be applied to single protein species analysis using fluorescent antibodies, as well as multiple species analysis using non-specific labels for all the proteins separated by electrophoresis or chromatography.
The most critical point of the technique is obtaining data reproducibility. I recommend keeping the same experimental condition as much as possible when measuring multiple samples. Visual demonstration of this technique will provide how to make each experimental step stable and reproducible. It will also show some uncommon steps in biochemistry protocols, such as the spin coating and washing of microscope cover slips.
To begin this procedure, prepare the lysing buffer and 1% tween 20 as outlined in the text protocol. Use sodium hydroxide to adjust the pH to 12. Mix 100 microliters of lysing buffer with 1 microliter of 1 molar DDT and 4 microliters of 50%.